पढ़ेंगे recombination in bacteria. What is recombination? Recombination means the transfer of genetic material from one bacterial cell to another is known as the recombination in bacteria. I hope that you understand the meaning of recombination bacteria. There are three types of recombination in bacteria. One is transformation, second one transduction, and third one is conjugation. Though let's discuss one by one. What is transformation? This is one transformation. A bacterial cell takes up foreign DNA from the environment. Uh, The bacterial cell takes the foreign DNA from the environment. The uptake of DNA molecule, the uptake of DNA molecule through the cell wall from the external surroundings, followed by stable incorporation into the recipient genome or replication as an independent plasmid. Transferring the pre-DNA, which is, let's leave this part. The steps of transformations. What are the steps of transformation in bacteria? Step number one, remove plasmid from bacterial cell. Remove the plasmid from bacterial cell. Step 2, isolate the gene of interest. Step 3, cut open plasmid with restriction enzyme. Leaves sticky ends. This 2% is sticky ends. Sticky ends, leave the sticky ends. This step number 4, insert gene of interest. Step number 4, insert gene of interest. Step number 5, insert the plasmid with recombinant DNA into a new bacterium. Step number 6, the bacteria reproduces itself, the plasmid and the plasmid, all descendants express the inserted genes. Now, this phenomena is called bacterial transformation. What is transformation? The addition of foreign deoxyribonucleic acid DNA into a bacterium. This is plasmid, a circular piece of DNA, and this is gene of interest, a competent cell, a cell that is able to accept DNA. A transformed cell, a cell that has new DNA. Transformation was discovered in 1928 by Frederick Griffith. Spelling is wrong. Here E. Frederick Griffith. Now, second is transduction. What is transduction? A virus or bacteriophage moves DNA from one bacterial cell to another is known as transduction. When certain provirus turn into virulent viruses whereby causing lysis of the host cell, they may carry with it segment of the host's DNA. Such virus particles carrying a segment of DNA from one host infect another host that is different from past. Latter may inherit some of the properties of former host due to the transfer of DNA segment through the infecting phage. What are the steps of transduction? Number one is reception. The cell senses a change in the environment. Number two, transduction. The signal is sent through the cell. Response, third step, there is a change in cellular behavior. This phenomenon of the transfer of genetic material from one bacterial strain to the other by the agency of virus is called transduction. Here, this is bacteriophage. Phage is insert phage DNA. This is phage DNA, and this is all is donor cell. The phage protein code this, a bacterial chromosome this. Uh, Step number one, a page infects the donor bacterial cell. Second step, page DNA and proteins are made. Page DNA and proteins are made. The bacterial cell chromosomes is broken down into pieces. All are broken down into pieces. Step 3. Occasionally, during page assembly, pieces of bacterial DNA are packed in a page capsid. Then, the donor cell lyses and release page particles containing bacterial DNA. Lyses, this is lysis part. And step 4, a page carrying bacteria DNA infect a new host cell, the recipient cell. New a new recipient cell, and followed by a recombination can occur producing a recombinant cell with a genotype different from both the donor and recipient cells. Conjugation. DNA gets transferred between two bacterial cells through a tube. That is conjugation. Conjugation is an exchange of genetic material within two cells that is in direct contact with each other by forming a temporary bridge-like connection between two cells. In conjugation, one cell acts as a donor and the another cell acts as a recipient. Bacteria exchange their own genetic materials by this process. It is termed as sexual reproduction of bacteria as two cells are involved and mate with each other for their exchange. This is tube called pilus, and this tube is connected with both the cells, and here you can see that uh, chromosome DNA is transferring from one cell to another. This is old donor pilus. This is new donor. A plasmid here also, and here also a plasmid.